So one of the first phylums we're going to look at are the porifera, which are sponges. And sponges, if you recall, lack tissues. They have no true tissues, but they are multicellular. They are actually made up of multiple different types of cells like choanocytes and amoebocytes, and these cells work together. So they're multicellular organisms, but they don't have any tissues. They also lack a nervous system. They do have genes, but they don't have nervous tissue. Most of these organisms, the sponges, are asymmetric. And so they're also most often asexual. They fragment or bud off new organisms. They can reproduce sexually as well. The sponge itself is hermaphroditic, and so it produces the sperm and the egg, and it releases it out into the water. And then it can release sperm to a neighboring sponge, which then can produce new fertilized eggs and create new sponges. So most of the time, the sponges will release their gametes, which come from amoebocytes and choanocytes. And then once they meet and a zygote forms, then the larvae that form become flagellated and they swim through the ocean. And it's not until they plant themselves and start to undergo their development that they become stationary or sessile adults. These sponges are so unique because they have an intracellular digestion, but they don't have a body cavity in there. So they have no true tissues, just these cells. So on the outside are these amoebocytes, and they get like hard skeleton kind of things. They secrete this calcium or silica. And then they also form these little spicules, and the spicules are like sharp little needle points sticking off of it, right? And then they also have these choanocyte cells, which have flagella on them, and the flagella beat, and it sucks the water in through pores. The water comes in through the pores carrying like small microorganisms that the cells digest and then shoot out through the top of the sponge. So you can see here that this sponge has an opening at the top, and that opening at the top is the osculum. This is where things exit. The little pores in the sides of the cells are where the water comes in and the organisms come in. You can see the amoebocytes are on the outside, and then it secretes that sticky substance and makes those spicules from the amoebocytes. And then the flagellated choanocytes are on the inside, beating their little flagella, sucking the water in and pushing it out the osculum on the top. Each of the choanocytes represents a colonial protist, the choanoflagellate. And so that's how we know that is where the original Animalia stem from, that line of protist. As it draws in the water, it can digest the organisms that come in through phagocytosis. So they just invaginate the cells and surround the food and then use it, and they can share it between them. So it is debated whether or not the ctenophores come before porifera, but it makes sense when you think about the law of parsimony or the KISS principle that the sponges come first because they lack any true tissues and are asymmetrical. When we look at the ctenophores, we start to see, oh wait, there might be something here that is different. They have these long tentacles with coloblasts that secrete a sticky substance. So as their tentacles are flowing in the water, they can catch prey that are swimming by. They have 8 rows of cilia on the surface, and those rows of cilia look like combs kind of like the ones that you comb your hair, but more like the ones that you use in DNA electrophoresis, right? The combs that form the gels. And they are just shooting little cilia, and that's their form of locomotion. This is the largest animal to use cilia to move through the water. They are hermaphroditic, so they have the male and female parts, and they can fertilize. And then they luminesce, so they're bioluminescent, and that means they can glow in the dark. They have like a glow in the dark fluorescing protein. They have a nerve net instead of a neural system, it's a nerve net. And so they do show a nervous type system or nerve cells, just not as expansive of a system as other animals have. And they experience radial symmetry. Radial symmetry is much more developed than, say, the sponges with an asymmetrical formation or body plan. The book does indicate that ctenophores came first, and they base that on the evolution and molecular data, so it might be true, but just looking at the law of parsimony, they would be a branch off of the same origin of the cnidaria. So when we look at the cnidaria, we can see these are organisms that have two germ layers, like we were talking about. They're diploblastic, but they still have radial symmetry. These are some of the simplest organisms with two germ layers, with true tissues, right? There are things like hydra, jellyfish, sea anemone, and even coral. It's really interesting because they have a gelatinous substance that's called the mesoglia, and it connects the endoderm and ectoderm, but they don't have that actual mesoderm, just the mesoglia, so kind of like a cytoplasm for the innards of the body, holding the body system together, if you will. Most cnidarians have tentacles, and the tentacles are used to get food. And they also have multiple body types. We can look here and see a very good depiction of a hydra. The tentacles are swaying all around, trying to pass the water around so they can get food into the system, right? You can see that they do have two layers. They have a cell layer of ectoderm and a cell layer of endoderm. And then they have this body cavity that they ingest things into. It's their digestive cavity. You can see here in the cross section that it's just a hollowed out middle section. So there's no actual entrance and exit. There's only two layers of cells and then this internal cavity. And so it's diploblastic and acollimate. There's no cavity formed on the inside. So as with the porifera, the sponges, there are multiple body forms, and they do have a sessile form. In cnidaria, the sessile form is a polyp, and like the hydra was a good example of the polyp form. They have that tubular body with the tentacles, and then the opening at the top is both a mouth and anus. It takes in the food and puts out the waste, all out of the same area. They have that non-cavity central section that digests their food, and they have a motile form, which is called the medusa, which is what we think of typically as a jellyfish, right? And we see that it can move throughout the water with those tentacles performing the locomotion part. The mouth is on the bottom and it's surrounded by the tentacles. But again, still the mouth and anus are the same. It's not like they form a body cavity when they change between their two body forms. Special cnidocytes are cnidarians that contain nematocysts, and these are little, almost like harpoons that jut out and they trigger and sting other organisms, and that's how they like stun their prey to eat them, and that's why jellyfish sting. They have these little nematocysts that pop out and kind of harpoon you with a chemical that burns and stings. Some of them are sticky instead and they just trap you in them. But these cnidarians do not have muscles and they don't have nerves. They don't have a mesoderm, so they don't have any of these other tissues. They only have their 2 germ layers. So it makes sense to think that these radiosymmetrical diploblastic organisms, the cnidaria, also were split into the ctenophores, which were the comb jellies, instead of them coming before the asymmetrical sponge. So when we leave these radially symmetrical organisms that are identical as long as you draw the plane through the center of the organism, we get into these bilaterally symmetrical organisms. And so there is a dorsal and a ventral side, a top and a bottom. There is an anterior and a posterior, the head and the anus ends. But they also have a plane of symmetry that slices the animal in half and has 2 identical sides. They're mirror images of one another. So the cnidaria and the ctenophores were radial, the porifera were asymmetrical, but now all other animals in Animalia kingdom are bilateral. The bilateral body plan has the dorsal and ventral upper and lower, anterior and posterior head and tail, and they are all not identical, but the right and left are the mirror image. And bilateral symmetry correlated to cephalization, and that makes sense. Now the anterior mouth end becomes the head, and animals typically enter and take sensory information in on a new environment with the head. So the head is where the eyes, the ears, the nose, the mouth are. So that's how they detect prey. It's how they respond to predators. It's how they see danger, where they might live. It's how they sense the habitat or the ecosystem around them. Our bilaterally symmetrical organisms develop in one of 2 ways. Either they have a protostome development or a deuterostome development. And the protostomes are, as they are cleaving in their blastula, they form a blastopore that becomes the mouth first. And it creates this spiral type of cleavage. Where a deuterostome, the blastopore becomes the anus first. And then it has a radial or symmetrical cleavage in that radial fashion where you could cut it through the center and see how that cleavage could form identical pieces. So when we look at our evolutionary depiction again, we can see those parazoa are the sponges, no true tissues. Then we have eumetazoa with true tissues. We have the radiata that have 2 germ layers, and then bilateria that have the 3 germ layers. And then we start to differentiate between protostome and deuterostome development. And we will see this gives rise to acoellomates, pseudocoellomates, and coellomates.